Welcome to Science Store online classes.
Don't forget to like our video and subscribe our YouTube channel for all the updates.
Welcome to all my dear students. Today I am taking biology class, and at that we are studying a new chapter, that is cell, the unit of life. In previous class we have studied the anatomy of the flowering plants, where we have studied different types of the anatomy of the different parts of the plants, right? Stem, root, leaves. Okay, we have differentiated the monocot and dicot plants. In this class we are going to study about the detailed structure of the cell. Okay, before that in our environment, in our surrounding, we can clearly differentiate the living and non-living things, right? The main difference between living and non-living things is, here living things show an important characteristic feature, living functioning. You can see alva yavedella irtade living things anta helbekadre it should move alva. Most of the organism will move from one place to another place except the plants. Okay, here the plants also show living character like growth alva. Growth ಒಂದು important character, and also food production, respiration. These are the different function of a living organism. But here non-living things will not do any of these function alva. ಯಾವುದೇ ಈ ತರಹದ ಫಂಕ್ಷನ್ ಅನ್ನು ಮಾಡೋದಿಲ್ಲ. ಹಾಗಾಗಿ ನಾವು living and non-living thing ಅಂತ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಮಾಡಲಿಕ್ಕೆ ಆಗ್ತದೆ. Here what is the basis for this living organism? Why they are doing this different functioning? Okay, here this different functioning is carried out by the cells, different cells. Okay, here the cells which carry out the different functioning of the living organism. Okay, and here you can see two different types of the organism based on the number of cells. Based on number of cells we can find two types of the organism. I have explained unicellular and multicellular. Okay, in unicellular organism a single cell will act as an independent organism. Here unicellular ಅಲ್ಲಿ ಏನಾಗ್ತದೆ, ಒಂದು ಸಿಂಗಲ್ ಸೆಲ್ ಏನಾಗ್ತದೆ, ಒಂದು ಲಿವಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ಆಗಿ ಆ್ಯಕ್ಟ್ ಆಗ್ತದೆ. ಲಿವ್ ಇಂಡಿಪೆಂಡೆಂಟ್ ಆಗಿ ಇರ್ತದೆ, ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಇಟ್ ವಿಲ್ ಕ್ಯಾರಿ ಔಟ್ ಆಲ್ ದ ನೆಸಸರಿ ಫಂಕ್ಷನ್. ಸಿಂಗಲ್ ಸೆಲ್ can carry out all the necessary function, but in multicellular organism the cells are made up of many number of cells. Here the living organism is made up of many number of cells, and each group of cells is confined to particular function. ಏನು ಇಲ್ಲಿ ಸೆಲ್ಗಳು ಗ್ರೂಪಿಂಗ್ ಆಗಿದೆ, ಆ ಒಂದೊಂದು ಗ್ರೂಪ್ಗಳು ಒಂದೊಂದು ಫಂಕ್ಷನನ್ನು ಕ್ಯಾರಿ ಔಟ್ ಮಾಡ್ತದೆ. ಅದೇ ಯೂನಿಸೆಲ್ಯುಲಾರ್ ಆರ್ಗ್ಯಾನಿಸಮಲ್ಲಿ ಒಂದೇ ಸೆಲ್ ಎಲ್ಲ ಫಂಕ್ಷನನ್ನು, ನೆಸಸರಿ ಲಿವಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮಿಗೆ ಬೇಕಾದಂತಹ ಅಗತ್ಯವಾದಂತಹ ಎಲ್ಲ ಫಂಕ್ಷನನ್ನು ಆ ಒಂದೇ ಸೆಲ್ ಕ್ಯಾರಿ ಔಟ್ ಮಾಡ್ತದೆ. ಬಟ್ ಮಲ್ಟಿಸೆಲ್ಯುಲಾರಲ್ಲಿ ಅದೆಲ್ಲ ಗುಂಪು ಕಟ್ಟಿಕೊಂಡು ಬೇರೆ ಬೇರೆ ಕೆಲಸಗಳನ್ನು, ಒಂದೊಂದು ಗ್ರೂಪ್ಗಳು ಒಂದೊಂದು ಕೆಲಸಗಳನ್ನು ಮಾಡ್ತದೆ. So these are the two different types of organism based on the number of the cells. Okay, next, in unicellular organism the main function of the unicellular organism is the main function of unicellular organism is they show independent existence. You can see the independent existence. Okay, here and also the cell perform the essential function of life. Here the independent cell will carry out or perform essential function of cell. These are the two important character of unicellular organism. Here they show independent existence. As I told, the independent cell will act as an individual organism and they perform essential function like respiration, digestion, then metabolism. All the function, life process is carried out within a single cell. Okay, respiration agirbohudu, digestion, absorption, then any body building, cell division, all this reproduction, all this carried out by a single cells. They perform essential function of cell, of life. Essential function of life, perform essential functions of life. Okay, here that is a main characteristic of unicellular organisms. And the structure, okay, any structure which is lower than any structure or the anything less than a complete cell, anything less than a complete structure of cell will not show any life process. It will not show any, does not ensure that independent living. Okay, here any incomplete cell, in simple words, any incomplete cells ಅಥವಾ ಯಾವುದೇ ಒಂದು ಸೆಲ್ ಇದು ಯಾವುದೇ ಒಂದು ಭಾಗ ಲಾಸ್ ಆಗಿದ್ರೂ ಅದು ಲೈಫ್ ಪ್ರೋಸೆಸ್, ಇಂಡಿಪೆಂಡೆಂಟ್ ಲೈಫ್ ಆಗಿ ಅಥವಾ ಗ್ರೂಪ್ ಆಫ್ ಲೈಫ್ ಆಗಿ ಪರ್ಫಾರ್ಮ್ ಮಾಡಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಎಲ್ಲ ಸೆಲ್ಸ್ಗಳ ಫಂಕ್ಷನ್ ಇಂಪಾರ್ಟೆಂಟ್. ಇನ್ ಮಲ್ಟಿಸೆಲ್ಯುಲರ್ ಇಟ್ಸ್ ಓಕೆ ಬಿಕಾಸ್ ದ ಅದರ್ ಸೆಲ್ಸ್ ಇಟ್ ಕ್ಯಾನ್ ಮೇಕ್ ಅಪ್, ಅದರ್ ಸೆಲ್ ಕ್ಯಾನ್ ಮೇಕ್ ಅಪ್ ದ ಫಂಕ್ಷನ್. ಬಟ್ ಯುನಿಸೆಲ್ಯುಲರ್ ಆರ್ಗಾನಿಸಮ್, ದ any incomplete cell is not show any independent existence of or independent living organism. They cannot act as a independent organism ಅಲ್ವಾ. ಯಾಕಂದರೆ ಎಲ್ಲ ಫಂಕ್ಷನ್ ಒಂದೇ ಸೆಲ್ ಕ್ಯಾರಿ ಔಟ್ ಮಾಡೋದ್ರಿಂದ ಎಲ್ಲ ಆರ್ಗನಲ್ಸ್, ಎಲ್ಲ ಪಾರ್ಟ್ಸ್ ಸೆಲ್ ಇದು ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿರ್ತದೆ. ಓಕೆ, ದ್ಯಾಟ್ಸ್ ವೈ ದ ಸೆಲ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಸ್ಟ್ರಕ್ಚರಲ್ ಆ್ಯಂಡ್ ಫಂಕ್ಷನಲ್ ಯೂನಿಟ್ ಆಫ್ ಲಿವಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮ್. ದ್ಯಾಟ್ಸ್ ವೈ ಸೆಲ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಸ್ಟ್ರಕ್ಚರಲ್ and functional unit of living organism. Here the cell is called as structural and functional unit of living organism. So here all the cells together will form a organism, multicellular organism, or single cell will act as independent organism. In any case, the structure and function of the cell is very important for to make a living organism. Okay, by this we can conclude that any structure, complete structure is necessary for the living, for the normal functioning of the living organism, and also all the functions. If the structure is proper, then all the function will be carried out by the cells. Okay, so here structural and functional unit of the living organism, cell, the definition of the cells. Here before that, in previous in older days there were no microscope, no well developed equipment to observe the cell. But here nowadays we can clearly see the different kinds of cells ಅಲ್ವಾ. ನಮಗೆ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಇದೆ, ಎಲೆಕ್ಟ್ರಾನ್ ಅಡ್ವಾನ್ಸ್ ಟೈಪ್ ಆಫ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಇದೆ. ಬಟ್ in olden days the cell was first identified by Antony van Leeuwenhoek. Okay, here the cell was first found out by Anton van Leeuwenhoek. Leeuwenhoek, Antony van Leeuwenhoek, who first discovered the cells. Okay, first discovered the cell, and also he started to develop the microscope. He is a Dutch scientist who is also called as father of microbiology. Microbiology anna first start madidava Antony van Leeuwenhoek in very olden days, in 18s, okay, 1800 years, okay, ಆ ಇಯರಲ್ಲಿ in 18th century, in 18th or in 17th century, he started about study about the microorganisms. So he is called as father of microbiology, who first found out the cell. Okay, cell anna first found ಮಾಡ್ತಾನೆ. And also he started to develop the okay, he started to develop the microscope anna first small ಒಂದು ಈ ಥರ like he placed a lens here by this one hook like structure. This was a very early microscope. He placed the organism in this and he observed through the lens ಮೂಲಕ ಅದನ್ನು ಅಬ್ಸರ್ವ್ ಮಾಡ್ತಾನೆ. By this he discovered the cells. Who first discovered, ಯಾರು ಫಸ್ಟ್ ಸೆಲ್ಲನ್ನು ಡಿಸ್ಕವರ್ ಮಾಡಿದ್ದು? ಆಂಟೋನ್ ವ್ಯಾನ್ ಲೆವೆನ್ಹುಕ್, who first discovered the cell. Later Robert Brown, who discovered the nucleus, which is a very important part of the cell, which contain genetic material alva. Nucleus is a very important part of the cell which carry out the necessary function, which stimulate or which guide the function of the cell alva. ಯಾವುದೇ cell function, ಯಾವುದೇ cell ಒಳಗೆ function ಆಗಬೇಕಾದ್ರೆ ಈ ನ್ಯೂಕ್ಲಿಯಸ್ ಫಂಕ್ಷನಿಂಗ್ ಕರೆಕ್ಟ್ ಆಗಿರಬೇಕು. ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ವಿಚ್ ಗಿವ್ಸ್ ದ ಇನ್ಫಾರ್ಮೇಷನ್, ಓಕೆ, ಅದು ಗೈಡ್ ಮಾಡ್ತದೆ, ಇನ್ಫಾರ್ಮೇಷನ್ ಅನ್ನು ಕೊಡ್ತದೆ. ಈ ಸೆಲ್ಸಲ್ಲಿ ಇಂಥ ಪರ್ಟಿಕ್ಯುಲರ್ ಫಂಕ್ಷನ್ ಆಗಬೇಕು ಅಂತ ಗೈಡ್ ಮಾಡೋದು ಯಾವುದು? ನ್ಯೂಕ್ಲಿಯಸ್. So Robert Brown, who first found out the nucleus. Okay, ಯಾರು ಫಸ್ಟ್ ಸೆಲ್ ಫೌಂಡ್ ಮಾಡಿದ್ದು? ಆಂಟೋನಿ ವ್ಯಾನ್ ಲಿವಿಂಗ್ಹುಕ್. and nucleus found ಮಾಡಿದ್ದು ಯಾರು? ರಾಬರ್ಟ್. not found, it is discovered. okay, you should know the difference between discovery and invention. discovery ಅಂದರೆ ಇದ್ದದ್ದನ್ನೇ ಹುಡುಕುವಂಥದ್ದು, ಇನ್ವೆನ್ಷನ್ ಅಂದರೆ ಹೊಸದಾಗಿ ಡೆವಲಪ್ ಮಾಡುವಂಥದ್ದು. ಡಿಸ್ಕವರ್ ಅಂದರೆ ಇದು ಆಲ್ರೆಡಿ ನ್ಯೂಕ್ಲಿಯಸ್ ಸೆಲ್ಸ್ ಎಲ್ಲ ಆಲ್ರೆಡಿ ಇದೆ, we are discovering. any plants also we are discovering, it is already existed. okay, we are discovering. that's why Anthony and Robert Brown, who discovered cell and nucleus respectively. Later on the development of the microscope alva, initially ಈ ಥರ ಸಿಂಪಲ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಇರ್ತದೆ, ವೆರಿ ಒನ್ ಲೆನ್ಸ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು. ನೆಕ್ಸ್ಟ್ ಸಿಂಪಲ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಡೆವಲಪ್ ಆಗ್ತದೆ, ನೆಕ್ಸ್ಟ್ ಕಾಂಪೌಂಡ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಡೆವಲಪ್ ಆಗ್ತದೆ. ಆ್ಯಂಡ್ ದ್ಯಾಟ್ ಲೀಡ್ಸ್ ಟು ದ gradual evolution or the invention of the microscope will lead to the complete and detailed study of the cells. okay, nowadays we can see the electronic microscope. Very important type of microscope where we can study the detailed structure of cell organelles, cell function, and also we can also study the structure of the viruses. okay, virus ಈ electronic microscope invention ಆದಮೇಲೆ ಅಂತೂ we can clearly study the structure and function of the cells. function ಮತ್ತು structure ಅನ್ನು ನಾವು detail ಆಗಿ study ಮಾಡಬಹುದು. and also we can also study the very small microscopic organism, even viruses or even the mycoplasma. any organism can be studied by using electronic microscope. ಆಯಿತಾ, ಎಲೆಕ್ಟ್ರಾನಿಕ್ ಮೈಕ್ರೋಸ್ಕೋಪಲ್ಲಿ ನಾವು ಯಾವುದೇ ಚಿಕ್ಕ ವಸ್ತುವನ್ನು ಡೀಟೇಲ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡಲಿಕ್ಕೆ ಆಗ್ತದೆ. ಸೊ ಅರ್ಲಿಯರ್ ಡೇಸಲ್ಲಿ ವೈರಸ್ ಅದರ ಬಗ್ಗೆ ಎಲ್ಲ ಜಸ್ಟ್ ಒಂದು they just told about the namings. virus ide, inta virus ide, just imagined and represent. but later on electronic microscope we can clearly we can study about that in detail ಆಗಿ ಸ್ಟಡಿ ಮಾಡಲಿಕ್ಕೆ ಸಹಾಯ ಮಾಡ್ತದೆ ಎಲೆಕ್ಟ್ರಾನಿಕ್ ಮೈಕ್ರೋಸ್ಕೋಪ್. ಬಟ್ ಇಂಥದ್ದೊಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್ ಇದೆ ಅಂತ ಆ ಓಲ್ಡನ್ ಡೇಸಲ್ಲಿಯೇ ಇವರೆಲ್ಲ ಸೇರಿ ಡಿಸ್ಕವರ್ ಮಾಡಿರ್ತಾರೆ. So nowadays we are studying our study becomes so easy. ಅಲ್ವಾ. ಅವರೆಲ್ಲ ಡಿಸ್ಕವರಿ ಮಾಡೋದ್ರಿಂದ ಇಂತಹ ಸೆಲ್ ಇದೆ, ಈ ಥರದ್ದು ಸ್ಟ್ರಕ್ಚರ್ ಇದೆ, ಅದರಿಂದಾಗಿ ನಮಗೆ ಈಗ ಸ್ಟಡಿ ಮಾಡಲಿಕ್ಕೆ ಸಾಧ್ಯ ಆಗ್ತುಂಟು. ನಮಗೆ ದಟ್ ಮೇಡ್ ಅ ಬೇಸಿಕ್ಸ್ ಫಾರ್ ಅವರ್ ಸ್ಟಡಿ ಇನ್ ಡೀಟೇಲ್ಸ್ ಅಬೌಟ್ ಸೆಲ್, ಮೈಕ್ರೋಬಯಾಲಜಿ ಅಥವಾ ಯಾವುದೇ ಬಯಾಲಜಿ ಪಾರ್ಟ್ ಆದರೂ ಈ ಬೇಸಿಕ್ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಎಲ್ಲ ನಮಗೆ ಹೆಲ್ಪ್ ಮಾಡ್ತಾ ಉಂಟು. ಓಕೆ, ನೆಕ್ಸ್ಟ್ ವಿಲ್ ಸ್ಟಡಿ ಅಬೌಟ್ ದ ಸೆಲ್ ಥಿಯರಿ. ನೆಕ್ಸ್ಟ್ ವಿಲ್ ಸ್ಟಡಿ ಅಬೌಟ್ ದ cell theory, important part of cell. Okay, before ನಾವೀಗ ಡೀಟೇಲ್ ಆಗಿ ಸ್ಟ್ರಕ್ಚರ್ ಆಗಿದ್ರೆ, ಬಟ್ ವಿ ಶುಡ್ ನೋ ದ ಹಿಸ್ಟ್ರಿ ಆಫ್ ದ ಸೆಲ್, ಹೌ ದೇ ಫೌಂಡ್ ಔಟ್ ಇನ್ ಅರ್ಲಿಯರ್ ಡೇಸ್. ಇಟ್ ವಾಸ್ ವೆರಿ ಡಿಫಿಕಲ್ಟ್. They even don't have any well developed equipment for the study of cell, but still they studied about the cell very it is a microscopic structure, ಅದರ ಬಗ್ಗೆ ಸ್ಟಡಿ ಮಾಡಿದ್ದಾರೆ. ಸೊ we should know the history, okay, how this cell the study of cells started. Okay, the study of cells or cell structure is called as cytology. ಅದನ್ನು cytology ಅಂತ ಹೇಳ್ತಾರೆ. The study of cell or function or structure of cells are called as cytology. Cyto means cell, logy means study. So cytology means study of the cells. Okay, next we'll study about the cell theory. Very important in cell theory. Initially in 1838 Matthias Sheldon Matthias Sheldon, who discovered, he is a German botanist, who is a German botanist who is a German botanist who discovered, who examined a large number of plants, at this time the microscope development was started. Okay, the microscope invention was started. So he started to study about the different plants. He examined different plants and he observed that each plant is made up of different kinds of cells or different types of cells. Here he found out that each plant is made up of different types of cells. Okay, and these cells grouped and made into, or they are grouped and they grouped and form a specialized structure called tissues. Okay, he found out that the plant is made up of different types of cells, and these cells which form a tissue, he found out different types of cells in plants. Okay, and these cells which will group together and form a tissues. Okay, so he found out that these cells which form a tissues, and this tissue will meant for the different function. ಅಲ್ವಾ, ಪ್ರೀವಿಯಸ್ ಅನಾಟಮಿಯಲ್ಲಿ ಕಲ್ತಿದ್ದೀವಿ. ಟಿಶ್ಯೂ ಏನು ಮಾಡ್ತದೆ, ಒಂದೊಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಫಂಕ್ಷನ್. ಮೀಸೋಫಿಲ್ ಟಿಶ್ಯೂ ಏನು ಮಾಡ್ತದೆ, ಫೋಟೋಸಿಂಥಸಿಸ್ ಅನ್ನು ಮಾಡ್ತದೆ. ವ್ಯಾಸ್ಕ್ಯುಲರ್ ಬಂಡಲ್ ಏನು ಮಾಡ್ತದೆ, ಇಟ್ ಈಸ್ ಅ ಟೈಪ್ ಆಫ್ ಟಿಶ್ಯೂ ವಿಚ್ ಪರ್ಫಾರ್ಮ್ ವಾಟರ್ ಆ್ಯಂಡ್ ಫುಡ್ ಕಂಡಕ್ಷನ್. ಅಲ್ವಾ, xylem phloem ಇರೋದ್ರಿಂದ ಫುಡ್ ವಾಟರನ್ನು ಕಂಡಕ್ಟ್ ಮಾಡ್ತದೆ. ಸೊ ಈಚ್ ದಟ್ ಟಿಶ್ಯೂ ಇಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಡಿಫರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ cells. Okay, I have gave the definition. Tissue is a group of cell which perform a particular function. Okay, they group together and perform a particular function, and they have a common origin. Alva, common origin annu hondirtade. So that group of cells may be similar in structure and function. They may be similar in structure as well as different structure, you can see. Okay, different irtade athava similar irtade. Okay, next we'll study later on. At the same time, Theodore Schwann in 1839, Theodore Schwann The Theodore Schwann, who started to examine, he examined the animal cell. He examined animal cell, who discovered he is a British zoologist. He found out the different types. He is a British zoologist who discovered the different types of animal cells, and while studying he observed that these animal cells are made up of only thin membrane, the outer delimiting membrane. They have a outer thin delimiting membrane, or outer membrane is thin. Okay, that's why this animal cell later that membrane is called as, on later date, that membrane is called as plasma membrane. So later that delimiting membrane or the thin membrane around the animal cell is called as plasma membrane. He found out that, he also studied the plant cell, and he differentiated that here the plant cell contains a special type of cell wall. Here in plants you can see the cell wall. Here the delimiting membrane is cell wall. You cannot see the delimiting membrane as a plasma membrane. Here you can see one more layer around the plasma membrane. Alli sa plasma membrane irtade hige. Here animal cell alli it is contain only plasma membrane, but in plant cell you can see the outer wall, that is nothing but the cell wall. So by this way he differentiated the plant cell as well as animal cell. ಅವನ ಸ್ಟಡಿಯಲ್ಲಿ ಏನು ಮಾಡ್ತಾನೆ, ಈ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಆನ್ ದ ಬೇಸಿಸ್ ಆಫ್ ಮೆಂಬ್ರೇನ್ and cell wall, presence and absence of cell wall, he classified the cells into animal cell and plant cell. ಓಕೆ, ಏನು ಮೇಯ್ನ್ ಆಗಿ ನಾವು ಹೇಗೆ ಪ್ಲಾಂಟ್ ಮತ್ತು ಆನಿಮಲ್ ಸೆಲ್ಲನ್ನು ಡಿಫ್ರೆನ್ಷಿಯೇಟ್ ಮಾಡ್ತೇವೆ ಅಂದರೆ ಸೆಲ್ ವಾಲ್ ಪ್ಲಾಂಟ್ಸಲ್ಲಿ ಇರ್ತದೆ, ಆನಿಮಲ್ಸಲ್ಲಿ ಇರೋದಿಲ್ಲ. ಹಾಗಾಗಿ ಆನ್ ದಿಸ್ ಹಿ ಡಿಫ್ರೆನ್ಷಿಯೇಟೆಡ್ ದ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಆ್ಯಂಡ್ ಆನಿಮಲ್ ಸೆಲ್, ಆ್ಯಂಡ್ ಹಿ ಆಲ್ಸೋ ಪ್ರಪೋಸ್ಡ್ ಅ ಸೆಲ್ ಥಿಯರಿ, ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಹಿ ಪ್ರಪೋಸ್ಡ್ a cell theory. He proposed a cell theory that all the plant and animal cells are composed of cells. All plants and animals. Here this Theodore Schwann, who proposed a cell theory. In this theory he concluded that the plants and animals are composed of cells, and they are the product and these are product of the cells and products of cells. And he concluded that all the plants and animals are composed of cells and they are product of cells. ಅಲ್ವಾ, ಏನು ಎಲ್ಲ ಪ್ಲಾಂಟ್ ಮತ್ತು ಆನಿಮಲ್ಗಳು ಸೆಲ್ಲಿಂದ ಮಾಡಲ್ಪಟ್ಟಿದೆ. ಆ ಸೆಲ್ ಅಲ್ಟಿಮೇಟ್ ಪ್ಲಾಂಟ್ ಮತ್ತು ಆನಿಮಲ್ ಬಾಡಿ ಫಾರ್ಮ್ ಆಗ್ತದಲ್ಲ, ದ್ಯಾಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಪ್ರಾಡಕ್ಟ್ಸ್ ಆಫ್ ಸೆಲ್. ಓಕೆ, ಅದು ಪರ್ಟಿಕ್ಯುಲರ್ ವೇಯಲ್ಲಿ ಅರೇಂಜ್ ಆಗಿ ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಟ್ರಕ್ಚರನ್ನು ಕೊಡ್ತದೆ. ದ್ಯಾಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ ದ ಪ್ರಾಡಕ್ಟ್ ಆಫ್ ಸೆಲ್. ಓಕೆ, ಹಿಯರ್ ಲೇಟರ್ ದೇ ಆಲ್ಸೋ ಫಾರ್ಮುಲೇಟೆಡ್ ಬೋತ್ ಥಿಯೋಡರ್ ಶ್ವಾನ್ ಆ್ಯಂಡ್ Matthias Sheldon, they formulated cell theory. Okay, idu cell hypothesis. Cell hypothesis. This is a cell hypothesis given initially given by Schwann, Theodore Schwann. Later the Sheldon and Schwann formulated cell theory. In the cell theory they modified this definition. Ee definition anna modify ಮಾಡ್ತಾರೆ. They concluded that ಹಿಯರ್ ಏನು ಆಲ್ ದ ಪ್ಲಾಂಟ್ಸ್ ಆ್ಯಂಡ್ ಎನಿಮಲ್ಸ್ ಅಂತ ಹೇಳಿದೆ, ಅನಿಮಲ್ ಸೆಲ್ ಅಂತ ಹೇಳಿದಾನಲ್ಲ, ಇಲ್ಲಿ ಏನು ಮಾಡ್ತಾರೆ, ಇದು ಶೆಲ್ಡನ್ ಆ್ಯಂಡ್ ಶ್ವಾನ್ ಗಿವನ್ ಅ ಸೆಲ್ ಥಿಯರಿ. ಇನ್ ದೇರ್ ಸೆಲ್ ಥಿಯರಿ ದೇ ಕನ್ಕ್ಲೂಡೆಡ್ ದಟ್ ಆಲ್ ದ ಲಿವಿಂಗ್ organisms are composed of cell and they are product of cells. Okay, and they are product of cells. Okay, this is a cell theory formulated by Theodore Schwann and ಮ್ಯಾಥಿಯಾಸ್ ಶೆಲ್ಡನ್. ಓಕೆ, ಥಿಯೋಡರ್ ಶ್ವಾನ್ ಮತ್ತು ಮ್ಯಾಥಿಯಾಸ್ ಶೆಲ್ಡನ್ ಏನು ಮಾಡ್ತಾರೆ, ಸೆಲ್ ಥಿಯರಿಯನ್ನು ಫಾರ್ಮುಲೇಟ್ ಮಾಡ್ತದೆ. ದಿಸ್ ಈಸ್ ಸೆಲ್ ಥಿಯರಿ ಫಾರ್ಮುಲೇಟೆಡ್ ಬೈ ಶೆಲ್ಡನ್ ಆ್ಯಂಡ್ Schwann. They formulated this theory. Okay, here all the living organisms are composed of cells and they are product of cells. Okay, later on 1855 Rudolf Virchow he found out that all the cells, while studying the cells in detail, he studied that the cells are formed from pre-existing cells. By the cell division, the new cells are formed from the pre-existing cells. Here the parent cell undergo division and forms two cells. Okay, they form two cells. Okay, and these new cells are formed from pre-existing cells. So he modified the cell theory by adding one more point. This is the one point. Okay, all the living organisms are composed of cells and they are product of cells. And also he added one more point for the cell theory is the cells are originated from pre-existing cells. All the cells are formed, all the cells arise from pre-existing cell. Okay, all the cells arise from pre-existing cell. Already cell irtade, adu divide agi new cell agtade. So that new cell again act as a parent cell, adu puna divide agi they multiply. This number of cell get multiplied. Okay, so this is by this way you can see the new cell formation. So he discovered that new cells formed from the pre-existing cell. So he added one more point in the cell theory. So the cell theory includes all the living organisms are composed of cells and product of cells, and they all the cells arises from pre-existing cells. These are the important two cell theory, important points of the cell theory. Cell theory ಎರಡು important point, very important. Okay, asked in your examination. Okay, next we'll move to the cell. The overview of cell. We can study the overview of cells. Okay, initially you have observed in your lower classes, you have observed the onion peel alva, onion peel matte human cheek cell alva, cheek olaginda cheek inda ನಾವು ಸ್ಕ್ಯಾಬ್ ಮಾಡಿ ಸೆಲ್ಸ್ ಅನ್ನು ಅಬ್ಸರ್ವ್ ಮಾಡಿರ್ತೀವಿ. ಹೇಗಿರ್ತದೆ ಸೆಲ್ಸ್? ಆನಿಯನ್ ಪೀಲ್ ಸೆಲ್ ಹೇಗಿರ್ತದೆ? ಕ್ಯಾನ್ ಸಿ ದ ಬ್ರಿಕ್ ಲೈಕ್ ಸೆಲ್ಸ್ ಅಲ್ವಾ, ಬ್ರಿಕ್ ತರ ಅರೇಂಜ್ ಆಗಿರ್ತದೆ ಇಲ್ಲಿ ಸೆಲ್ಸ್ ಲೈಕ್ ದಿಸ್ ಅಲ್ವಾ. Can observe the arrangement of the cells like this. ಓಕೆ, ಆನಿಮಲ್ ಸೆಲ್ಸ್ ಯೂಶಲಿ ದೇ ಆರ್ ಚಿಕ್ ಸೆಲ್ ಈ ಥರ ಇರ್ತದೆ. ಸೆಂಟ್ರಲ್ here in plant cell you can see the definite structure of the cell because of the presence of this cell wall. ಓಕೆ, ರಿಜಿಡ್ ಸೆಲ್ ವಾಲ್ ಇರೋದ್ರಿಂದ ಅದು ಡೆಫಿನೆಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನು ಹೊಂದಿರ್ತದೆ. ಇಲ್ಲಿ ಇಟ್ ಈಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಡಿಲಿಮಿಟಿಕ್ ಮೆಂಬ್ರೇನ್ ಈಸ್ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಅಲ್ವಾ. ಔಟರ್ ಮೆಂಬ್ರೇನ್ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಮಾತ್ರ ಇರ್ತದೆ, ವೇರ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಇರ್ರೆಗ್ಯುಲರ್ ಶೇಪ್. ಓಕೆ, ಸಮ್ವಾಟ್ ಇರ್ರೆಗ್ಯುಲರ್ ಶೇಪ್ ಇರ್ತದೆ, ಕರೆಕ್ಟ್ ಎಕ್ಸಾಕ್ಟ್ ಸ್ಕ್ವೇರ್ ಅಥವಾ ರೆಕ್ಟ್ಯಾಂಗಲ್ ಶೇಪ್ ಇರೋದಿಲ್ಲ. ಓಕೆ, Here you can see observed under microscope. Usually we will observe these cells and study the structure of cell by using microscope. You have observed the plant, the animal, this onion peel leaf, where you can observe the cell with the cell wall. Rigid cell wall irtade. So animals with the plasma membrane. By this way you can clearly differentiate the plant cell and animal cell. It is a onion peel is a good example for the animal plant cell. Plant cell ige olle examples agirtade. We can clearly differentiate the plant cell and animal cell, and in each cell we can find a dense membrane containing organelles in dense membrane containing structure that is called as nucleus. Nucleus or prominent or the dense. Okay, it contains dense or compactly arranged structure and it is covered by a membrane, thin membrane. That structure we'll call it as nucleus. Here inside the nucleus you can see the chromosome. I have told the nucleus is an important part of the cell which guide the various functioning of the cell alva. Ee nucleus eno guide ಮಾಡ್ತದೆ cell idu various function ಅನ್ನು ಹೇಗೆ ಅಂತ ಹೇಳ್ತೇನೆ. Nucleus which contain chromosome in turn contain genetic material. This genetic material is very important. It will code for particular protein mRNA ಮೂಲಕ mRNA ಅಂತ ಒಂದು ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತದೆ, ಅದರ ಮೂಲಕ ಯಾವ ಪ್ರೋಟೀನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬೇಕು. ಯೂಶಲಿ ನಮ್ಮ ಫಂಕ್ಷನ್ ಎಲ್ಲ ವೇರಿಯಸ್ ಫಂಕ್ಷನಿಂಗ್ ಈಸ್ ಕ್ಯಾರಿಡ್ ಔಟ್ ಬೈ ದ ಪ್ರೊಡಕ್ಷನ್ ಬೈ ದ ಫಾರ್ಮೇಷನ್ ಆಫ್ ಡಿಫರೆಂಟ್ ಕೈಂಡ್ಸ್ ಆಫ್ ಪ್ರೋಟೀನ್ ಫಾರ್ಮೇಷನ್ ಆಗೋದ್ರಿಂದ, ಬೇರೆ ಬೇರೆ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಫಾರ್ಮೇಷನ್ ಆಗೋದ್ರಿಂದ ನಮ್ಮ ಸೆಲ್ ಫಂಕ್ಷನಿಂಗ್ ಮಾಡ್ತದೆ. ಸೊ ಇದೆಲ್ಲ ಫಂಕ್ಷನ್ ಅನ್ನು ಯಾವುದು ಕಂಟ್ರೋಲ್ ಮಾಡೋದು? ನ್ಯೂಕ್ಲಿಯಸ್ ಇಂದ ಈ ಇನ್ಫಾರ್ಮೇಷನ್ ಅಥವಾ mRNA ಅಥವಾ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆಗದಿದ್ರೆ ಯಾವುದೇ ಸೆಲ್ಯುಲಾರ್ ಫಂಕ್ಷನಿಂಗ್ ಆಗೋದಿಲ್ಲ. ಸೊ ದ ಮೆಂಬ್ರೇನ್ ಬೌಂಡ್ ಆರ್ಗನೆಲ್ಸ್, ಡೆನ್ಸ್ ಮೆಂಬ್ರೇನ್ ಬೌಂಡ್ organelle present inside the cell is called as nucleus, and this nucleus which contain chromosome that in turn contains genetic material, and this genetic material as DNA, deoxyribonucleic acid. Okay, DNA full form enta deoxyribonucleic acid. So this is called as genetic material that helps that maintain the cell. Okay, here in some of the cells the nucleus is not the genetic material is not covered with the membrane. Is here if the nucleus is not covered with the membrane, here the nucleus covered with the membrane, then that type of cell we'll call it as eukaryotic cell. Na helidene previous class alli the difference between eukaryotic and prokaryotic cell. Here the nucleus is covered with the membrane. Okay, idara nucleus genetic material idre, this is a genetic material, chromosome irtade idralli. Here this is covered by a membrane. Usually this is covered by a membrane like structure. Okay, this type of nucleus containing cells are called as eukaryotic cells. Sometimes in what happens, there is no membrane around the genetic material or the nucleus. Okay, there is no genetic membrane around the genetic material, and that type of cell which contains this genetic material without a membrane, that type of cells we'll call it as prokaryotic cell. That we'll call it as prokaryotic cell. That type of cell we'll call it as prokaryotic cell, primitive type of cell where the nucleus is not covered by membranes. Okay, that is the main difference between eukaryotes and prokaryotes. I have mentioned in the previous classes. Okay, next we'll move to the next we'll study about the cytoplasm. Here all the cells contain a inner fluid matrix. Okay, all these cells contain a inside to cell you can find a fluid matrix. Okay, fluid matrix, that we'll call it as cytoplasm. Fluid part of the cell is called as cytoplasm, and it occupies the volume of the cell. Maximum volume of the cells are occupied by cytoplasm. Okay, all these organelles are suspended ಅಥವಾ ಅಲ್ಲಿ ತೇಲ್ತಾ ಇರ್ತದೆ. ಈ ವಾಟರ್ ಕಂಟೆಂಟ್ ಇರ್ತದಲ್ಲ, ಸೆಮಿ ಸಾಲಿಡ್ ಫ್ಲೂಯಿಡ್ ಥರ ಇರ್ತದೆ ಸೆಲ್ ಒಳಗೆ, ಅದರಲ್ಲಿ ಈ ಆರ್ಗನಲ್ಸ್ ಎಲ್ಲ ತೇಲ್ತಾ ಇರ್ತದೆ, ಸಸ್ಪೆಂಡ್ ಆಗಿ ಇರ್ತದೆ. ಸೊ ದ್ಯಾಟ್ cytoplasm is very important, where it is a main arena of cellular activities. Main arena of cytoplasm is main arena of cellular activity. Here the cytoplasm is the main arena of cellular activity because all the chemical reactions, okay, all type of biochemical reactions or metabolic process is taking place in the cytoplasm region. Cytoplasm region alli a chemical reaction agudrinda, so that is the main arena or the area of the cellular activity. Cellular activity aglikke adu main area. Okay, cytoplasm ee region alella ಈ ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ ಆರ್ ಬಯೋಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ಆಗ್ತದೆ. ಸೊ ದಟ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಸೈಟೋಪ್ಲಾಸಮ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, ಆ್ಯಂಡ್ ಇಟ್ ಆಲ್ಸೋ ಹೆಲ್ಪ್ಸ್ ಟು ಬಿಕಮ್ ದ ಸೆಲ್ ರಿಚಿಡ್ ಅಲ್ವಾ. ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಸ್ಟ್ರಕ್ಚರ್ ಮಾಡಲಿಕ್ಕೆ ಹೆಲ್ಪ್ ಮಾಡ್ತದೆ ಈ ಸೈಟೋಪ್ಲಾಸಮ್. ಮತ್ತು ಸೆಲ್ ಎಲ್ಲ ಆರ್ಗನಲ್ ಸಸ್ಪೆಂಡ್ ಆಗಿರ್ತದೆ. ಸೈಟೋಪ್ಲಾಸಮ್ ಇಲ್ಲಾದರೆ ಹೇಗಿರ್ತದೆ, ಗಾಳಿಯಲ್ಲಿ ತೇಲ್ತಾ ಇರ್ತದೆ. ಇವನ್ ದ ಏರ್ ಇಸ್ ನಾಟ್ ಪ್ರೆಸೆಂಟ್, ದ ಸೆಲ್ ಬಿಕಮ್ ಶ್ರಿಂಕ್. ಓಕೆ, ಫ್ಲಾಸಿಡ್ ಆಗ್ತದೆ ಸೆಲ್ಸ್, ಮತ್ತು ಆರ್ಗನಲ್ಸ್ ಎಲ್ಲ ಶ್ರಿಂಕ್ ಆಗಿ ಅದರ ಫಂಕ್ಷನಿಂಗ್ ಮಾಡಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ, ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ ಆಗ್ಲಿಕ್ಕೆ ಆಗೋದಿಲ್ಲ. ಸೊ ಸೈಟೋಪ್ಲಾಸಮ್ ಈಸ್ ಅ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾರ್ಟ್. Okay, important part. We can see both in prokaryotic and eukaryotic cell. Okay, both prokaryotic ಮತ್ತೆ eukaryotic ಅಲ್ಲಿ matrix containing or the fluid matrix containing area will call it as cytoplasm. Okay, and this outer membrane will call it as plasma membrane, and around this plant cell you can see the cell wall. Okay, cell wall irtade. So this is about the important part of the cell, and along with that you can see in higher cells, in eukaryotic cells you can see the membrane bound organelles. Okay, membrane bound structures. Okay, inside the cell you can see membrane bound structures. The membrane bound structures usually called as organelles. These membrane bound structures inside the cells are called as organelles. Usually these organelles are present in eukaryotic cells. Different types of organelles present in eukaryotic cell, and the prokaryotic cell will lack membrane bound organelles. Okay, your prokaryotic cells alli untalla ee membrane bound organelles alla present irudilla. Can you give different examples for membrane bound organelles? Okay, we will study the endoplasmic reticulum, mitochondria, Golgi complex, You have studied about this alva, study cell 9th standard ಅಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡುವಾಗ ನೀವು ಪಾರ್ಟ್ಸ್ ಆಫ್ ಸೆಲ್ಸ್ ಅನ್ನು ಕಲ್ತಿದ್ದೀರಿ, ಆರ್ಗನಲ್ಸ್ ಎಲ್ಲ ಆರ್ಗನಲ್ಸ್ ಅನ್ನು ಕಲ್ತಿದ್ದೀರಿ. ದೀಸ್ ಆರ್ ದ ಡಿಫರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಸೆಲ್ ಆರ್ಗನಲ್ಸ್. ಗಾಲ್ಗಿ ಕಾಂಪ್ಲೆಕ್ಸ್, then chloroplast, then vacuole, okay, then anything endoplasmic reticulum, mitochondria, Golgi complex, chloroplast, vacuole, lysosomes, micro bodies, lysosomes, micro bodies. These are the different kinds of membrane bound organelles present within a eukaryotic cell. Only eukaryotic cells alli ee membrane bound organelles irtade, ade prokaryotic cells alli membrane bound organelles irudilla. Okay, should remember. And here in prokaryotic cell they lack membrane bound organelles. Okay, instead of that they have some modification that we are going to study while studying the prokaryotic cell. Okay, and also here these lysosome, here the prokaryotic, the non-membrane bound organelle is called as ribosome. Ribosome is ribosomes do not contain any membranes around their structure. They do not contain any membranes. They simply made up of RNA, ribonucleic acid ಇಂದ ಆಗಿರ್ತದೆ ಮತ್ತು proteins ಇಂದ ಆಗಿರ್ತದೆ. ಇದರಲ್ಲಿ membranes ಇರುವುದಿಲ್ಲ. These ribosomes are present both in prokaryotic and eukaryotic cell. They are present in both prokaryotic and eukaryotic cells. Okay, these ribosomes ಎರಡರಲ್ಲಿ ಸಹ ಇರ್ತದೆ. And also some in eukaryotic organism within some organelles. Okay, cell ಒಳಗೆ ಇರ್ತದೆ usually, within some organelles ಅದರೊಳಗೆ ಸಹ you can find ribosomes. For example, chloroplast present in plants, right? ಎಲ್ಲಿರ್ತದೆ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್? ಎನಿಮಲ್ ಸೆಲ್ಲಲ್ಲಿ ಇರ್ತದ? ನೋ, ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಪ್ರೆಸೆಂಟ್ ಓನ್ಲಿ ಇನ್ ಪ್ಲಾಂಟ್ ಸೆಲ್. ಅದರೊಳಗೆ ಇನ್ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಇನ್ಸೈಡ್ ದ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ವಿಚ್ ಈಸ್ ಔಟರ್ಲಿ ಕವರ್ಡ್ ಬೈ ಮೆಂಬ್ರೇನ್. ದಿಸ್ ಕ್ಲೋರೋಪ್ಲಾಸ್ಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಮೆಂಬ್ರೇನ್ ಇಂದ ಕವರ್ ಆಗಿರ್ತದೆ. ಇನ್ಸೈಡ್ ದಿಸ್ ಯು ಕ್ಯಾನ್ ಸಿ ದ ಸೆಪರೇಟ್ DNA ಆಸ್ ವೆಲ್ ಆಸ್ ರೈಬೋಸೋಮ್ ಮತ್ತು DNA ಇರ್ತದೆ. ಆ್ಯಂಡ್ ಆಲ್ಸೋ ಇನ್ ಮೈಟೋಕಾಂಡ್ರಿಯಾ. In mitochondria you can see the presence of genetic material as well as ribosome ಮತ್ತೆ genetic material ಇರ್ತದೆ. It is cell ಒಳಗೆ ಈ organelles ಇರ್ತದೆ. ಈ organelles ಒಳಗೆ ಸೆಪರೇಟ್ ಆದ ಒಂದು ಸ್ವಲ್ಪ DNA ಮತ್ತು ರೈಬೋಸೋಮ್ಗಳು ಇರ್ತದೆ. Okay, this is about the important basic difference between prokaryotic cell and eukaryotic cell, and also the overview what and all the cell, the particular or the basic cell should contain these type of parts. ಯಾವುದೆಲ್ಲ ಇರ್ತದೆ? ನ್ಯೂಕ್ಲಿಯಸ್ ಇರ್ತದೆ, ಮೆಂಬ್ರೇನ್ ಬೌಂಡ್ ಯುಕ್ಯಾರಿಯೋಟಿಕ್ ಸೆಲ್ ಆದರೆ ಮೆಂಬ್ರೇನ್ ಬೌಂಡ್ ಆರ್ಗನಲ್ಸ್ ಇರ್ತದೆ. ಅದೇ ಪ್ರೊಕ್ಯಾರಿಯೋಟಿಕ್ ಸೆಲ್ಸ್ ಆದರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ವಿದೌಟ್ ಮೆಂಬ್ರೇನ್. Okay, and also it do not contain membrane bound organelles, but they contain ribosomes. Okay, ribosomes ಇರ್ತದೆ. These are the different basic study of the cells. In next class we will study about the size, shape and their activities. Okay, and later we will study prokaryotic cells, okay, with important parts of the prokaryotic cell, and next we will move to the eukaryotic cell. Thank you all my dear students.
Don't forget to like our video and subscribe our YouTube channel for all the updates.